Good morning, everyone. Welcome to Grand Rounds. My name is Alex Harris, and I'm one of the Grand Rounds co-directors, along with Drs. Aditya Lati and Rebecca Muley. I want to start by thanking the area leads and chair again for their ongoing support, sponsoring the food before and after Grand Rounds out of their personal funds during this hopefully coming to an end austerity period. There are still a few spots left for the trainee lunch, so trainees, please take advantage of the opportunity to meet with today's speaker. This week, we have two important town halls. Today at 2 p.m., there's a virtual Attune town hall. This is specific for the basic neuroscience laboratories. It will walk users through the animal use protocol submission process, and it's mandatory for all animal users, and attendance will be tracked. An email went out this morning with the details. Tomorrow at 10 a.m., there's an in-person clinical town hall. Dr. Josh Gordon will be leading this in the multipurpose room. It's the first of 3 town halls planned for the summer. It's open to all Department of Psychiatry and NISP clinical faculty and staff across New York State, faculty practice, and NYP. If you'd like more information or details about either of these events, please reach out to Iris Delgado. We would like folks to please nominate internal departmental faculty who would be good speakers for the upcoming academic year. Please send us an email to grandrounds@nisp.columbia.edu. Next week's Grand Round speaker will be Dr. Philip Gehrman, Professor of Clinical Psychology in Psychiatry at the University of Pennsylvania. The title of his talk is Sleep, Circadian Rhythms, and Mental Health. Before we introduce today's speaker, a reminder that if you're participating via Zoom, please post questions using the Q&A feature, not the chat. I'm joined today online by Dr. Rebecca Muley, who will forward questions to me for our speaker to address. We encourage trainees to consider asking a question, and we'll prioritize trainee questions in person and online. Please remember to fill out your CME surveys after the talk if you would like CME credit for today's lecture. There's a QR code. There will be time for the speaker, I promise, on the table outside for those in person, and the link will be posted on the chat on Zoom at 12:15 p.m. It's now my great pleasure to introduce today's speaker, our very own Dr. Matthias Quick. Probably most of you already have come into contact with Matthias, and certainly all of you have benefited from his efforts. That's because Matthias has leadership positions in virtually every aspect of what makes this place run. He's chair of the NISP EHS committee, the safety committee, the DCM operations. He's the right to know officer, the chemical hygiene officer, and a member of the space committee, the emergency management team, among others. I personally would like to take this opportunity to thank him for all that work. But today we're here to benefit from Matthias in a different capacity as an accomplished biophysicist and molecular neuroscientist. Matthias started out in Germany, where he obtained his bachelor's and master's in biology and his PhD in microbiology and membrane transport at the University of Osnabrück. During his graduate work under the supervision of Dr. Heinrich Jung, he developed several tools that aided in the functional and structural characterization of sodium-dependent symporters. After graduation, Dr. Quick did postdoctoral work on the human sodium glucose transporter in the laboratory of Dr. Ernest Wright at UCLA, where in addition to functionally characterizing this protein with electrophysiologic methods, he established the functional expression of recombinant sodium glucose transporter in bacteria. Dr. Quick joined the Center for Molecular Recognition in 2003, where together with Dr. Jonathan Javich, he studied the structure and function relationship of members of the neurotransmitter sodium symporter family. And if I'm reading his CV correctly, he first came to RFMH in 2005. In 2008, he was appointed to the faculty of the Department of Psychiatry. His work has substantially contributed to our better understanding of the molecular events underlying sodium-coupled transport processes, which, for context, play a key role in everything from nutrient absorption to kidney function to neurotransmitter removal. The title of his talk is Elucidating Neurotransmission at Atomistic Level. Please join me in giving him a warm welcome.
Thank you, Alex, for the very nice introduction, and thank you all for showing up today. It's a pleasure to be here. I think what Alex mentioned, I heard some other reports, and I usually stand here to give some safety updates, but today I'm talking about science. I have some scientific background, and I'm talking about transport. As you can see, there's a transporter on the display here. What Alex missed in his introduction, I also served in the army. I was in the transportation battalion. So that's why I learned to drive trucks, and everything related to transport is basically what I do. So title today is Elucidating Neurotransmission at Atomistic Level. Conflict of interest statement. In short, I have no conflict of interest because there's no financial interest here. What are the learning objectives for today's talk? Obviously, that's focusing on the neurotransmitter sodium symporter family, or the NSS family, also known as the SLC6 family, based on different family arrangements, basically. The objectives are describe the molecular underpinnings of neurotransmitter transport. Secondly, list the energetic requirements associated with neurotransmitter transport. And last, discuss the complexity of neurotransmitter transport and the action of drugs. So this is what I'm trying to do today. Today's talk is split up into 3 parts: some background, some recent work that I started with Jonathan back when I joined the division of molecular therapeutics. And then also some new work that we started recently and will be continued in the near future. So what about transport? So this is a slide that I saw on my kids' high school preparation for college, and it shows a cell, and it's all very complex and dense. What's missing, however, in my opinion, is this one here, my favorite topic, a transport protein or a membrane protein sitting in a membrane, because this is essential to communicate with the environment, with other cells. So this is basically missing. And in all this complexity, the same complexity applies to the brain. So basically, transport proteins or membrane proteins are essential for any life on Earth. Going back to my grad student work in Osnabrück, I worked in the microbiome department, and I was working with bacteria. So the topic, and this gives you some background as to why I'm doing what I'm doing and how I do this, relates to a bacterial cell, and my target was a proline transport system. So the idea was that bacteria transport proline along with a proton-coupled symport mechanism. The simple fact was, if you add some acidity to the solution, the proline transport goes up. And this matched the idea back in the days when there was the lac permease, which was a proton-coupled lactose symporter that was analyzed by and maybe even abused by Ron Kabeck, who happened to be the supervisor of my supervisor. So he was in Los Angeles as a postdoc in Ron's lab. He came back. I was his first undergrad and then grad student. So he was basically biased, I may say. So basically the idea is that you have, sorry, this was the wrong button, that basically protons drive lactose uptake into bacteria. The protons come from the respiratory chain and also for the action of the ATPase. Basically, this drives uptake of lactose. So the big picture back in the days, like late 80s, was all bacterial transport systems are dependent on protons, whereas mammalian systems are more dependent on sodium. So the sodium glucose transporter was one big family. The other part was lac permease. So I worked with this protein, and yes, I saw when I dropped the pH, protein uptake goes up. But then something didn't add up, and I figured out that there is a different system in the cells, which is called the sodium proton antiporter. So when you add protons, dropping the pH, it transports sodium out of the cell. So this then happens that the sodium is used by this protein, the protein transporter, PAD-P, and it turns out this is a sodium-coupled protein transporter. There was this Japanese group, mid-late 80s, making a huge publication list where they have a kinetic model of how protons do this, how do that. But in reality, PAD-P, the system is a sodium-coupled protein transporter. And what's even more tricky, the affinity for sodium is about 35 micromolar. So have you ever worked with a system that has such an affinity? That's why I'm working today with systems that are low in sodium. So if anybody tells me I'm using a buffer and I titrate with sodium hydroxide, I'm getting a little nervous, I would say. So my buffers, as I have tried them, can be reduced in to a sodium contamination of about 2 micromolar. So when you work with a system that has such a high sodium affinity, everything that you touch with your bare hands or so is so high in sodium, you can't distinguish any specifics. So basically, I tried to explain why I was doing this, and I'd like to tune out the noise. I focus on one specific protein at a time, and I'm trying to assess the molecular details of this particular protein, and then generate the mechanistic model of function. And in the end, science is a team sport, so I'd like to add one piece of a puzzle, and then the people who work with me can rely on this piece of information being accurate rather than having lots of information that's not adding to the picture. Some background. So as I mentioned before, some people I talked in the past, I don't think anybody is in this audience, who believe that that transport across the membrane is by diffusion. This is not the case. So there are different types. There is passive transport, is diffusion. There's facilitated diffusion, channel mediated. There's active transport, and there's also group translocation. This is very rare, so we ignore this here. But if you talk about diffusion in channel mode, it basically shows that you have a protein in the membrane that's opening its gates, and then through those gates, depending on the specificity of the protein, you have flux of substrates, ions, compounds, whatever. And to make this a very simple model, this came from a slide from Chris Miller when he presented in Wisconsin, and the student came up and told him, look, this is all very abstract. This is what I can really get familiar with. So basically the idea is once the gate is open, all the cows rush out. The cows go rushing out as long as there are cows on my right side, and if there's space on the left side. And then why do I show this? Well, this is where I grew up. Basically, it's close to home. But it always conveys the message. So the other part I'm trying to talk about is the active transport component, and this is where I'm focusing on the NSS, on secondary active transport. This is basically where there is no opening on both sides. There's basically a gating mechanism where you have opening on one side, the other side is closed, you have an occluded state, and you have also the release of the substrate to the other side, but not to this side because the gate is closed. So there's always an alternating access from one side of the membrane or the other, never at the same time. Of course, there's a similar mechanism. As you can see here, the cow, again, is the substrate. This here is our energizer. Let it be the sodium in our system. So you can see it takes effort to get the cows through, and there's only one cow going one direction and only when the gate is open. The other cow in the back doesn't rush through. So there's a huge difference between principles of channels and transporters, and also the turnover numbers are different. In a channel, you have about 1 million ions can pass per second. In a transporter, like the cow here, is by maybe 1 to 50 per second. So there's a huge difference in the timing of the events. This also gives you an idea of how effective a protein can be. It's called the alternating access model. And then I simplified this here in the beginning, but it's more complex. It's not just one co-translocated ion, there may be more. And it's even more complex. And let's not talk about this, but I just want to mention this here. Transporters can also have channels incorporated. So you have dual function of transporters and channels. So this brings us to the NSS family. And you know this scheme, basically, you have the presynaptic neuron, action potential causes the release of neurotransmitters from the vesicles in the synaptic cleft, where they interact with the receptors on the postsynaptic neuron. The clearance of the cleft is then basically made or established by different proteins, the dopamine transporter, norepinephrine transporter, serotonin transporter. Those are the members of the SSS or SLC6 family. The energy is provided by the sodium gradient through the action of the sodium-potassium ADPase. So sodium is one of the driving members or ions of this process. And you can see that all the three systems are listed here show sodium, and then there's substrate, and then there's also chloride. So these transporters are basically sodium, chloride dependent. In the case for serotonin transporter, you have also potassium antiport listed in 1 or 2 publications. Why it is so interesting, particularly for this audience, because they're the primary target for therapeutic and illicit drugs. So this has been studied for 4 decades, I would say, starting with Saul Snyder and Jonathan joined the lab later, and then Baruch Khanna in Israel, where he started to isolate proteins and try to focus on the specific function of a protein. Cloning was done in the mid-90s, and then basically following this new development, we have much more information on how these transporters work. However, what was missing was the structure. So these things cannot be isolated easily out of human brains or any brain, basically. So what people have come up with, they used model systems. And one of the model systems that was very successful is called LuT. It's a transporter from a thermophilic bacterium that was crystallized in Eric Go's lab in 2005. And this showed basically an amazing resolution of 1.65 angstrom. You could see the overall structure. You could see one molecule of leucine bond in the central binding site. You could see the exact location of two sodium ions shown in cyan and blue here. And then two helices are shown in red and green. Basically, this is the active part of the protein where you have the binding of the substrate. If you look at the overall structure, it's a very dense structure with no opening. There's no open pore on this or that side. Just to make the point, this is outside of the membrane. This is inside the membrane. So this is basically what came out in 2005, was an amazing development to see such a structure. And then just to give you an overview of the same structure, you have 12 transmembrane domains that span the membrane. You have two inverted repeats here of these elements. You have TM1 and TM6, as I showed in the previous slide. You have a leucine in the center bound here. You have one of the sodium ions in the sodium 1 side, also in the central pocket, which is actually coordinated by the carboxy group of leucine. And then you have a second sodium ion, which is a little bit off, which is more, or was proposed to be more regulatory. What do you do if you have a single snapshot of a structure? You make a model, and basically what I showed you before with the alternating access model, you have access of leucine and the sodium from one side. You have this occluded state, which represents the structure. And then you have this release of substrate and sodium to the inside. This is a very simple model. Well, amazingly, shortly after the first crystal structure of LuT was published, there were two other papers by the same group, by Eric Go and then also by Daneng Wang's group, where I have LuT co-crystallized with tricyclic antidepressants. So there's one is clomipramine, the other one is imipramine. And what's interesting, you have a very similar structure. You have leucine bound here. You have the drug sitting on top of the leucine. And when you look at the structures with and without the drug, they look almost identical. So what you would expect, the drug would impair function by making major changes to the structure, which is not the case. So all this came up. We were wondering, how is this happening? And I'm interested in the function, so we tried to understand what happened. So we look at the paper and see the functional description of LuT. And when I look closer, the turnover number of LuT is in the order of 0.0004 per second. Remember, I told you before, a channel has a turnover number of 1 million per second. A transport has a turnover number of 1 to 50 per second. This here is like 10 orders of magnitude slower. Something is really wrong. Or let's say it the other way. For leucine to go from the outside to the inside, it takes 35 minutes. It doesn't look very efficient. So this was very suspicious. And then we tried to repeat the purification and all the functional studies. It didn't really work. And I'll tell you in a second why. But if you look at why is there leucine in the structure, so LuT was cloned, expressed in bacteria, and it was cells were cultivated. Then they prepared the membrane vesicles, getting rid of the cytoplasm, only focusing on the membranes in the absence of leucine. They solubilized the protein in the absence of leucine. They purified LuT in the absence of leucine. They polished with a size exclusion chromatography in the absence of leucine. They performed the crystallization in the absence of leucine and ended up with a structure of 1.65 angstrom, which is bound to leucine. Where does this come from? Well, simply here. It comes from the cells, and if you wash the cells at least 4 times in the absence of sodium, then you can get rid of the leucine that's bound there. But the problem was, if you just do this preparation without the specific care, you end up with a leucine-bound structure. And why is there leucine? Because the affinity is so high. Leucine binds with an affinity of about 40 nanomolar to LuT. It's just very tricky to get rid of leucine, which compromised many subsequent functional studies. And then what I also have to tell you, leucine is not the only substrate. Leucine is actually the worst substrate in terms of transport. There are other amino acids, methionine, isoleucine, valine, and alanine. Alanine is the best transported substrate, so it should be maybe, as opposed to LUT, LRT maybe. But you name it what you see the first time. And this is a trend that I see more and more often. Proteins are called by their, I don't know, preferred substrate, or the substrate was found one way or the other, and then it turns out these things do something totally different. They have additional functions I mentioned. They have channel activities, and the substrate specificity changes. So based on the structure we had already available, we worked with Harold Weinstein's group at Weill Cornell with Lei Shi. Basically what he has done, he used a structure of LUT and tried, there we go, the wrong one again, sorry. And basically what he tried to do, he tried to pull out leucine from the S1 side. We call this the S1 side, which is the central binding side. He tried to pull it out, and what you would usually expect, you pull, and eventually it loosens up and shoots out. What he did, however, or the results he got, leucine was always stuck in this pocket here. So when he pulled from here, up there, or all the way out, it always ended up here. So he came up with the idea there's probably a second binding site for leucine. And this matched with some functional studies that I performed at the same time. And this was very helpful that we developed a SPA binding assay, a scintillation proximity assay, where you have a scintillant that's covered by copper. And if you have radioactive compounds in the medium, you have no energy transfer here. Only if you have a protein, in this case, a his-tag protein like LUT, it binds to the copper-coated beads. And then once it's held in close proximity, bound radioisotopes elicit some kind of a light signal. So you can detect binding directly without any separation steps. You can have a perfect equilibrium binding approach. And that's what we have done. So we used LUT and performed binding, saturation binding of tritiated leucine. And you can see here we used 4 picomole of LUT, and you can see for wild type, we have about 8 picomole of leucine bound, which 8 divided by 4 makes it 2. So basically 2 molecules of leucine are bound per molecule of LUT. If you look closer on this slide here, so there are 2 residues are highlighted, which is F253 and L400. L400 is a residue that goes right into the proposed S2 site, and F253 is sitting right on top of the S1 site. When we mutate these individually, we basically lose binding of one side. So for the L400C variant, we lose binding to this side. We only maintain binding to this side. And if we do the F253 mutant, we lose binding here, maintain this one here. So basically, this tells us there are 2 binding sites that are involved in this whole process. Then you could argue, well, but the sodium sites are very close. So we measured also sodium-22 binding, and we show here this wild type, and this is the F400 C mutant. Basically, you maintain sodium binding, and believe me, for the F253 mutant, binding is the same. So we maintain a stoichiometry of 2 sodium per LUT, which matches the 2 sodiums identified in the structure. And an amazing thing that also happened, given the high affinity of leucine for LUT, we could have something called trapping. And this trapping was a very nice feature, which I haven't seen in any other protein before or after, but it helped solve many, many of the mechanistic details. And I just summarized this here, which became the cover of Molecular Cell in 2008. We have basically shown here, we have sodium binding to the empty transporter. We have then, in the presence of sodium, we have binding of leucine. We have then still an exchangeable state. This here is then a doubly occupied LUT, where you have leucine in the S1 side. Sodium is also here. Leucine binds to the S2 side. Then the next step is dissociation of sodium 2 comes off first. And then after, once you have leucine in the S2 side, leucine in the S1 side, and sodium 1 leave together. We tried to publish this in Cell, but Cell didn't like it because Cell insisted on having the entire cycle. Basically, you have this open transporter here. You have binding of sodium and substrate, doubly occupied. You have a change in conformation from the outside to the inside, and you have release of substrate, and then you have a transition from the inside facing to the outward facing state. what we showed basically was just this partial cycle here. Cell wanted this one, so we ended up in molecular cell. Wasn't bad either, so I'm not complaining. So a couple of years go down, and what we see, LUT is not the only member with the structure. So we have the NSS family, SLC6, we have LUT, we have SERT, we have DAT. We have in the SSS family, we have the glucose transporters, we have PADP, the one I grew up with basically, and we have other families, and they all share the same structure. And this one shows all the proteins I worked with. So before I knew they had the same structure, or even after, so I'm basically familiar with these guys. So I mentioned before, when I tried to assess the function of the protein, there was always an issue with octoglucoside. And by chance, more or less, we tried working with a protein, a LUT variant, which has this E290 mutated to an S. We tried to figure out the chloride or proton dependence of the protein. And this is a different story, but what's more important here is basically that octoglucosides inactivates LUT, as I mentioned before. But in this structure, you can clearly see that octoglucoside molecules stick from the outside to this S2 binding site in the two monomers in this dimer structure. We have additional octoglucosides sitting on the outside, but what's important, these two sit right into the S2 binding site, and this prevents the function of LUT. And if you look at the original structure, it was published by Eric O's group in 2005, there is indication of density in those binding sites, which would explain why we couldn't find the function, because octoglucoside sits in this binding pocket. Following this stretch of publications, there was the era of debate. So there's a two-side model, and then there was also the one-side model, basically seeing is believing. If you see only a structure with one leucine bound, it's a one-side model. Everything that we have published was not accepted. So we had to find another way to argue our way through. So if you look at this one, this here is another protein called MHST, a multi-hydrophobic substrate transporter, also bacterial origin. And if you look at the comparison of this S2 binding site region, this is LUT. So we have leucine 29, which is rather short. MHST has the tryptophan 33, which aligns with tryptophan 103 and tryptophan 51 in HSERT and DDAT, respectively. So we tried to understand, why is this different in LUT? Maybe it's the exception to the rule. We then tried to mutate this tryptophan 33 in MHST, and what we can see when we measure binding, here I did the math for you before. So basically here I show the moles of tryptophan bound per moles of MHST. So we end up with a stoichiometry of 2. So basically 2 tryptophan molecules bind to MHST in wild type. If we mutate W33 with a cysteine or an alanine or leucine, we maintain the binding stoichiometry. This is just to give you some oversight of the whole structure. So this here is tryptophan bound. This here is 253 in LUT. This is F230 in MHST. This residue I104 is sitting also in the S1 site. S2 site residues is isoleucine 111 and F381, which corresponds to leucine, for leucine 400 in LUT. And then there's this residue W33. As I showed you before, there's no effect on the binding affinity. Also, the transporters with the mutations transport almost normally. So we tried something that was done in Arthur Carlin's lab with Miles Acabas. We had a discussion before about him. So basically what we tried to do is we used a cysteine labeling approach by putting a cysteine at the position of this W33. So basically what we have here is a thio group sticking out, and here we have this disulfide, and then what we have in the end, we break this bond, and this adds up here, adding substance, some bulky elements in this binding pocket. And then we performed tryptophan binding studies. So for wild type, in the absence of MTSEA, we see 100%. Why is this? Because wild type MHST has no single cysteine, so there is no interaction of a thiol reagent in the protein, and there's no cysteine. If we replace W33 with a cysteine, we see in this position that MTSA binds and prevents binding to about 50%, which indicates or suggests at least that this pocket is then too spacious for tryptophan to bind. Sorry. In this case, we mutate an S1 residue with a serine. This causes no binding of MTSA because there's no cysteine. If you have a combined of the S104S, sorry, the isoleucine 104S and the W33C, which means MTSEA can bind here. This one is impaired by the mutation. We lose entirely tryptophan binding. And in this side, we see no binding, but we see the reduction of the binding from 100 to 50% because of the impairment of this binding site. There's two binding sites. And when we add the double mutant and add also MTSA, there's no effect because the binding site is already impaired, which means this binding pocket is still intact. We then perform transport measurements in the presence of the MTSEA. We used wild type and we used the W33C mutant. And in the absence of any compound, we have 100% uptake by wild type. W33 mutant has a similar level of uptake. If we add MTSEA to this mutant here, we lose entirely the uptake. If we add sodium, we still lose the uptake. If we use tryptophan alone, we use entirely the uptake. But if you incubate in the presence of MTSEA, sodium and tryptophan, the two subsets of MHST, we basically block MTSEA access to this pocket because we entirely fill up this pocket with sodium and with tryptophan, preventing the steric access of MTSEA to this site. And then the other part we have performed with MHST, we have obtained in collaboration with the Nissen lab in Aarhus, Denmark, a structure of MHST. And again, we saw similarly, very much the same as we saw with LUT. We have an S1 occupied site with tryptophan here. The S2 site is open. Continued work with Leichy's group at NIDA. Basically predicted a binding site for tryptophan in this S2 site. And then I performed binding studies. And I mentioned before that octoglucoside was a bad detergent for LUT. For MHST, it turns out that DDM, which was used for this crystal structure, is the bad detergent because if you replace DDM with DM, decyl maltoside versus decyl maltoside, we see a binding stoichiometry of 2. In DDM, we see a binding stoichiometry of 1. We then introduced nanodiscs where we have MHST incorporated into a lipid bilayer that's held together by a membrane scaffold protein or a bed protein. And you can see what this looks like. So you have purified MHST. This is the membrane scaffold protein, the gold and blue stuff here. And then in the nanodisc, we have the combined bed protein and MHST sitting. And when we measure binding, and it doesn't matter if the protein was purified in DDM or in DM, once it's incorporated back into the membrane lipid, we see full activity, meaning we have 2 binding sites accessible. And there's one more part for MHST where it's very helpful. So what I want to tell you is that Ron Kabik introduced the cabecosomes. So when you make membrane vesicles out of bacterial cells, you can have 2 versions. One is an inside-out vesicle. The other one is a right-side-out vesicle. And you can basically produce the right-side-out vesicles or cabecosomes by osmotic lysis and the inside-out vesicles by pressure treatment. So you have an intact bacterium, you add pressure or you use lysis, and you get vesicles that basically have whatever you want in them, depending on the buffer choice. And they have the membranes. The inside is facing outside, or in the other case, you have the outside facing outside. This picture was basically taken by Sam Silverstein back in the 60s here at Columbia when Ron was working with Sam. He told me the story recently. And then again, there's this whole thing that Ron Kabik came up with. There's this one paper, the kamikaze approach to membrane transport in 2001. I showed you this one before. protons coupled, or protons drive uptake of lactose in regular cells. You can also have the inverse reaction where you have lactose drives protons. Because it's a coupled process, basically one cannot happen without the other one. But you can also drive the opposite transport direction, which basically, if you load the vesicles with lactose and in the presence of low pH, you can also drive efflux of the system. Basically, the transporter is working both directions. The gradient is the only determination of what direction the transport is working. So to test this out, we had a collaboration with Scott Blanchard at Weill. Now he's in St. Jude. And there was some discrepancy in terms of forward and backward reaction. So we tried a different system, which is a glycine transporter, which is proton coupled, shown here. And we produced inside-out vesicles and right-side-out vesicles, either those that express MHST and also control cells, just to focus on the glycine transporter. As you can see here, in the case for the right-side-out vesicles in cyan and white, basically you get full activity. The inside-out also produces almost full activity, but if you add lactic acid, which feeds into the electron chain, and basically in the inside-out vesicles produces a flip of your proton gradient, you entirely block this process. So basically, as long as you have a proton gradient from the outside to the inside, you can drive uptake in those vesicles. When we measured used the same vesicles basically from this preparation and measured uptake of leucine for MHST, leucine, tryptophan, isoleucine, phenylalanine are all transported substrates for MHST. So in this case, we used leucine for consistency with other studies. But what you can see here is basically only the right-side-out vesicles produce active transporters, whereas in the inside-out vesicles, you have no uptake. Which means that transport by MHST, and we propose for all the NSS also, is only in one direction. You only have the sodium driving gradient going from the outside to the inside. You will never have the opposite. Which brings us back to the mechanistic model. This was basically based on von Kabek's reversibility of transport, which didn't get us into cell. But if you look carefully, I think this is what's happening. And this is exactly what we described back in the 2008 molecular cell paper. So there is no reverse step for the NSS family. So, and then I told you we have this structure of lutein in 2005. We added the 2008 molecular simulations and functional studies that we have two binding sites here. And the group of Eric was always proposing a single site model. And in 2021, they produced a structure of H-third by cryo-EM. And guess what you found? Serotonin in the S1 site and serotonin in the S2 site. And if you merge the two, the S1 and the S2 site, whether it's for lutein, is it leucine or alanine for this matter, or if it's in H-third for serotonin, the binding sites S1 and S2 basically match perfectly. What you do not see here is the sodium ions are missing because of the low resolution. That's why you need crystallography versus cryo-EM, where you have different resolution levels and can see certain elements of the protein. So basically what I showed you before was all focusing on the bacterial homologs of this family. And now I want to switch to the serotonin transporter. But just to give you some idea about, I tried to describe the molecular underpinnings of NSS transport and list the energetic requirements. And then just a short summary here. The NSS members possess two substrate binding sites termed the S1 and the S2 site. And there are also two sodium sites. And their allosteric interaction regulates transport. Sodium is the driving cation that energizes transport. Lithium, while binding to the sodium sites and enabling substrate binding, does not promote transport in lutein. So this is a study we published. Published this, Jonathan. Hey, actually, we need to publish this. And then also what I mentioned before, there's chloride symport in the mammalian systems. And you find chloride co-transport is essential in the eukaryotic NSS members, and antiport, proton antiport is the charge balancing mechanism in the bacterial members, where you have basically chloride goes along with the substrate in the eukaryotic ones. And then in the bacterial members, you have protons coming out, where you have substrate and sodium coming in. So there's a certain way to balance the charge distribution during transport. So so we have established a expression and purification protocol for SERT, and then we, as I mentioned before, we reconstitute the protein into liposomes. We use this method where we perform liposomes, destabilize them with detergent, extract detergent by biobeads, and end up with liposomes that have usually one protein per liposome. And when we measure uptake of serotonin, we can see a clear signal over background. So this is serotonin uptake, use 3-deated serotonin here, and in control liposomes where there's no protein, there's basically no uptake. We then try to analyze the ionic requirements. So in the presence of sodium chloride, which is the same as this one here basically, and the inside of the liposomes had a pH of 7.5, no other addition. If we then replace sodium chloride with sodium iodide, we lose some activity. So this chloride is actually really important to stimulate full uptake activity. And if we then also add potassium to the liposomes, trying to mimic this condition, I would have expected a much higher signal, but this doesn't really work out. So I'm careful what this really means if potassium is required here. Just to give an idea how this whole thing looks, we have purified HSERT. We have this in liposomes, protoliposomes reconstituted. We also have this incorporated to nanodisk, as I mentioned before. And then there's the membrane scaffold protein in both cases, where we just run this. But basically, we can incorporate HSERT in liposomes and nanodisks, and they maintain full activity. When we tried binding, what I'm showing here is basically, this is HSERT wild type. This is HSERT cis light construct, which is basically a construct of HSERT, which has the exposed cysteines replaced with other residues. And the idea is to perform EPR studies in the future in collaboration with Hassan Mekharab in Vanderbilt. The idea is basically here that we want to make sure that the protein is fully active. We have then basically performed those binding studies in detergent. We also performed these with nanodiscs. We have SERT reconstituted in a nanodisk. We use the CLC version, and we can see there's full activity. If we perform the binding in the presence of an excess of cold serotonin, in the presence of cocaine, amphetamine, or nisoxetine, basically binding is completely blocked. And we also performed amphetamine binding, and we can see that in wild type and in the CLC construct, we see nice binding of 3-deated amphetamine. MHST, which was used as a control, there is no binding of amphetamine. We also tried to understand the electrogenic properties of HSERT, and we performed solid-supported membrane electrophysiology measurements. We used this Surfer N1 device, which is basically a device where you have a quick exchange of solutions. And this is a scheme using a different protein. Basically, what the machine measures is the change in membrane potential. So this is basically at a steady state level, your membrane potential is what it is. And then if you add a substrate, which is being transported into the liposomes, the pentane SERT, you have a change in the membrane potential, and you have either efflux of negative charges or influx of positive charges, or the other way, you have charge flux. And depending on the signal, you have an upward or a downward trend. In this case, this means that positive charges enter the liposomes or negative charges exit the liposomes. You basically have an activating solution present. You have a change in membrane potential. You reach a steady state level here again. And then when you change the buffer to the non-activating condition, you have a return to the baseline. When we perform this with HSERT in liposomes, we can see that when we add some serotonin, you see a small signal, which is not surprising if you consider sodium maybe accompanied by chloride. So you have only if it's 2 sodium per 1 chloride, only 1 positive charge going in, which is basically what we can see here. Then we tried cocaine. Cocaine is known to block uptake of serotonin. However, if you look at the currents here, there's a huge signal. And if you perform this with amphetamine, we see an even bigger signal. So this means whereas cocaine and amphetamine are known to block the activity, something else is happening here. So there's some electrogenic event happening, and I suspect this may be sodium being rushing through. When we perform this in control liposomes without H-cert, we see no signal at all. Next step, we performed transport, and this is basically the same as I mentioned before. We have wild type, we have the CLC construct. In the absence of cocaine, amphetamine, or mesoxetine, we basically see nice uptake. All these block. And then the other thing we performed amphetamine, tritiated amphetamine uptake in control liposomes, where there's no uptake, and in H-cert wild type or CLC expressing liposomes. And we see a robust signal of amphetamine uptake. Amphetamine is known to mediate the efflux of serotonin. And what we have done here in this experiment, we have performed uptake in liposomes using radiolabeled serotonin. And then over time, 50 minutes, we have basically reached a steady state level where we have, in the presence of hot serotonin on the outside, it accumulates in the protoliposomes. And at this time point, we add either cold amphetamine or cold serotonin. And this is our 100% level in this next figure here. So then we waited for 50 minutes in the presence or absence of amphetamine and or serotonin. And then basically at this point, we would have started here. If we perform this experiment in the absence of any of these two, we have a little bit of increase. We're basically staying at the steady state level. If we add cold serotonin, there's basically no change in the accumulation of serotonin in the liposomes. But if we add amphetamine here, we lose about 50% of the accumulated serotonin, which is consistent with the idea of amphetamine-induced efflux of amphetamine. And what we propose now in this new model is that given the fact that amphetamine is transported, that we have amphetamine-induced efflux of serotonin, which is basically an amphetamine-serotonin exchange mechanism. So this is something we try to understand more in detail in the future. And the fact that we have now the protein ready and waiting for some other technical difficulties to be solved in addition to funding issues. But so this is the next step that we try to do. For this purpose of the learning objectives, basically I showed you that this is repeat basically NSS members have two substrate binding sites and two sodium sites, and this is the allosteric interaction that regulates transport. The experimental conditions, choice of detergent, for example, can obscure the integrity of the S2 site, thus affecting the activity measurements. Mutations in the S1 and the S2 site abolish transport. The same is also true for the sodium sites. If you mutate residues in the sodium binding pockets, you lose binding as well as uptake activity. The NSS transport cycle is unidirectional, which is opposing the old standing mechanistic idea of having both passes are possible. We have established H-cert activity measurements either using purified protein or reconstituted into nanodisk or liposomes. And then also what we found, H-cert activity transports amphetamine, and then cocaine blocks uptake of neurotransmitters. Amphetamine elicit reverse transport or efflux, and the hypothesis is that the antipode mode is triggered by amphetamine. And this leads me to thank you and everybody who participated in this work. So we have Jonathan, obviously, who took me in at some point 20 years ago. And then we have a couple of people who worked here. And currently we have Audrey, I think she's in the audience. Eva is on vacation this week. So they've been working on many projects, particularly focusing on the cert. We have Scott Blanchard working with the single molecule setup. At Weill Cornell, we have Harold Weinstein, George Galashvili, and Michael Levine. Formerly, Leishi was in Harald's group. Now he's at NIDA. Sebastian was in his group, and then Ara was also working on the MHST story. Hassan working on the EPR, focusing on the third story. I'd like to thank the Grant Runs Committee, Alex, Adi, and Rebecca. Karen was very helpful in organizing getting to this point, and who was also very helpful was Simon Williams, getting me in here yesterday and trying to fix my PC to Mac disaster. So thank you, Simon, for this one. And as always, for all the shows he's helping with. And those are the funding sources. And I thank you for your attention.